Hello, hope I'm audible. I think we can start.
My voice is breaking, ma'am.
Yeah, just a second. Am I audible now? Is it fine?
Yes, ma'am.
Okay. Okay. So I hope my presentation is also visible. We can start. If anybody wants to join later, they can join. Okay. So welcome to week 8, experimental biotechnology live session. Uh, this course is offered by Professor Vishal Trivedi in SWAYAM NPTEL, and I'm one of the TAs, Sumati from University of Hyderabad. This is my email ID. So this week's assignment covered basically cell culture, media techniques, centrifugation, uh, separation, etc. So based on that, we will discuss few topics which will help you solve the questions. So coming to the first topic, it was regarding the media and what is the main buffering system in the media. So in mammalian cell culture, buffer system is basically used to uh, maintain the stability in pH, maintain a constant pH so that the cells do not get affected due to the changes in the pH, either due to utilization of the nutrients from the media or due to uh, the secondary metabolites that are secreted by the cells after a while. So all these things can change the pH of the media. So we need a buffer system which will make uh, the pH in the media stable and constant, and it can help in viability of the cells. Excuse me. Yeah. So pH stability is critical because even small deviations can impair the growth, metabolism, and viability of the cells. And any changes in these parameters will definitely impact on the experiment that you have planned with your cells. So, the proper health of the cell is very important. The main buffer system used for this is bicarbonate CO2 system. So the buffer system is a combination of a component that is present in the media, which is sodium bicarbonate, and the CO2 gas that will be supplied to the incubator. So when it comes to cell incubators, we have two uh, types basically based on the carbon dioxide. One is CO2 incubator, which has a constant supply of CO2, and one is a non-CO2 incubator, which does not require any special addition of CO2 gas injection in it. So the non-CO2 incubators are basically used for microbial cultures or like mostly for bacteria, yeast, etc. Whereas CO2 system is used for the mammalian cell culture. So the bicarbonate from the medium and the CO2 pumped into the incubator, both together function as a buffer system for the cells. So how it works is the bicarbonate buffer system. The medium contains the cell culture medium in which you grow the cells. It contains sodium carbonate. And inside the incubator, there is constant supply of CO2 gas, which will then dissolve into the medium. So when CO2 gas dissolves into the medium, which contains sodium bicarbonate, so there is formation of carbonic acid, which then dissociates into H plus and HCO3 minus ions. So CO2 plus water, that is present in the medium, will give you H2CO3, which will then again dissociate into H plus and it's CO3 minus. So there is generation of the H plus ions here. So when there is growth of the cells and then the secondary metabolites are released, the media tends to move towards the alkaline system. So this buffer system helps maintain it in the neutral or in the physiological pH of 7.2 to 7.4. So this maintains the pH around 7.2 to 7.4, that is the physiological range. And the incubator atmosphere, uh, the standard supply of carbon dioxide is 5% carbon dioxide in all the cell culture incubators, unless you need to change for your experimental conditions. For normal growth, 5% carbon dioxide is standard and is carefully controlled to balance this system. So most commercial medias like DMEM or RPMI are formulated with the bicarbonates as the main buffer. Other than this, phosphate buffer is not usually for the pH. The phosphate buffers like either the monosodium or monobasic or dibasic phosphate, they are sometimes added into the media, but they help resist minor shifts in the pH, but they are not used as the main buffer system. The buffering capacity is effective for this between 6.8 to 7.2. So this is close to the phosphates pKa. So this will not help you maintain the physiological pH of 7.2 to 7.4. So this will only be maintained by the bicarbonate and the carbon dioxide system. However, the phosphate concentration is kept relatively low in the media to avoid precipitation with calcium and magnesium. So all these are salts, so they can get precipitated. So this might disturb the cell adherence or the cell morphology, physiology, everything. So these are all usually kept at a minimum, relatively low concentrations. So phosphate buffering is not supplementary, is supplementary when it comes to this range, but not used as a primary system. Then other than that, the other options were ammonium buffer system. So this is not for physiological buffering. Ammonium can act as a buffer, but in cell culture, ammonium is usually a metabolic waste from the glutamine metabolism. So this will make the pH more alkaline, and accumulation of ammonium usually causes toxicity to the cells and pH instability. So it is not used, it is not a correct option for buffer system. Then protein buffer system. So proteins, especially albumin, can buffer H plus in amino acid side chains, but it is not in the medium. So this is also not correct option for the buffering system. The serum free medium is defined to have low protein content, and even serum containing media do not rely on proteins for buffering. So this is weak and it cannot buffer efficiently. So the bicarbonate carbon dioxide buffer system is primary and intentional buffer system in the mammalian cell culture. Since it closely mimics the physiological conditions, it works effectively in the pH range cells, which require a range of 7.2 to 7.4, and it is controlled via incubator carbon dioxide levels together. Then the other buffer systems may contribute slightly, but they are not reliable and they are not used as a buffer system to maintain pH in cell culture in vitro. So this was about the buffer system of bicarbonate and CO2, how and why it is used in the mammalian cell culture in the media for maintaining the physiological pH. While other buffer systems can be used. So other than this, these other options were completely not correct. They cannot be an alternative in any way. But other than bicarbonates, HEPIS is a buffer which is used to maintain buffering conditions later on during experimental protocols, maybe while isolation of a particular organelle or while isolation of proteins, etc. Because it's a very good buffer system. But even this is not used in the cell culture medium. So, bicarbonate is physiological and non toxic, provides a metabolic substrate. It's precise in pH control and requires sealed CO2 incubator to prevent pH shift. HEPIS does not require a CO2 incubator because it is used in the buffers which are used later on after cell extraction. So strong buffering capacity in physiological pH range but however can be toxic to some cells at higher concentrations. It is not natural component of the mammalian physiological environment and may interfere in some experimental assays. So only depending on the experiment you want to use or the isolation protocols, you can use the HEPAS. Otherwise, the bicarbonate CO2 is the only system that is used while growing the cells. So other traditional buffers used for short-term incubations have low buffering capacity and they are not reliable for long-term pH stability. So this is a not so clear picture, but this shows the changes in the pH in the cell culture media. So the neutral or the physiological pH is somewhere between 7.2 to 7.4 and is constantly maintained by the CO2 and the bicarbonate. If somehow the buffer system is not able to maintain the physiological pH, then we have a shift in it where you can see it getting either alkaline or acidic in nature. So this is 7.4, this is 7, this is 6.5. So this here is getting acidic. So this happens when there is some sort of contamination in the media. So this is also a way to check whether your system is free of contamination or not. If you the media can always indicate if there is any contamination. So how can you visualize this? Even though the bicarbonate is there, it is still not visible to the naked eye. So we add something called as phenol red. So phenol red is a pH indicator. So this is pink at physiological pH, pinkish reddish pink. And if it goes, so this is actually how it looks in physiological pH in a reddish pink or an orange shade. So when the pH shifts to alkaline environment, then it becomes pink or magenta. So here it is somewhere 7.8 or 7.9. And when the pH shifts to acidic, it becomes light orange to yellow. So this color change will help you know whether your pH is being maintained in your system or not. So this is again an equatorial representation of the reaction. This is your CO2 when dissolved in water becomes H2CO3 and then you have your ions H plus and bicarbonate. So, when there is an increase or a decrease in the pH, these uh, the H plus ions will help balance the buffer system depending on whether the H plus is required or not. So, if it is getting more alkaline, this reaction will go forward and increase the H plus concentration. And if it gets more acidic in nature, then this reaction goes this way and it gets more basic in nature, and that's how it can balance the pH. Then there is also something called a reserve bicarbonate where you have the sodium and the bicarbonate present, which also is in an equilibrium in terms of sodium ion and the sodium bicarbonate substrate as well. So this is basically how your buffer system works with the help of an indicator which will help you visualize the change in the buffer pH. So the most commonly used indicator is phenol red. So coming to the next, this is also based on the pH and your indicator. So the question is an assertion and reason. So the assertion is that the color of the mammalian cell culture media turns yellow over time, and the release of H plus, the reason is the release of H plus ions released by the metabolism of cells reduces the pH of the medium. The low levels of the pH turn the medium yellow due to the pH indicator. So both A and R are true, and R is the correct indicator. So you can see that over time, not only during contamination, also depending on the metabolic waste, like if there is more alkaline substrates that are being released into the media, it will make the media alkaline, but mostly the H plus ions that is getting released into the media due to the metabolism will change the media to acidic. So this is a correct explanation of the assertion. So phenol red is a pH indicator dye, as I mentioned. It's not a nutrient. If you can use a media even without phenol red, uh, if you don't have to really care about the pH or changes, or if you're quick enough to replenish your media before it if there is before it gets acidic. So you there are also media available without phenol red, and also if you want to do certain assays in which phenol red might interfere with your uh, detection, if it's a colorimetric detection, or if you want to do some sort of imaging where you think the phenol red might interfere, you can definitely buy a media which is free of phenol red. It is equally good as your phenol red media, but the only thing is you cannot visually see if there is any change in the pH. So phenol red is not a nutrient, so it's not like a mandate in the media for growth, but it helps in detection of pH change. It is included in most commercial mammalian cell culture media like DMEM, RPI, or MEM at around 15 mg per liter concentration. So, if the pH range is less than 6.8, the phenol red will be yellow in color, and the media also will be yellow in color. So, that shows that it's too acidic, and excess of carbon dioxide, high lactate, contamination, or overgrowth. These could all be the reasons for this state. So, if the media is in a pH range of 7 to 7.4, which is the optimum range, it will be orange or red, reddish orange. So, this is physiological and healthy medium. And if it is greater than 7.4, more at 7.6 and higher, so the media will turn pink and then purple. So this suggests that the media is too alkaline, and there is low CO2 present, and there is poor buffering system, and uh, prolonged cell culture is taken place. So this is how you can interpret based on the different colors. So this is the chemical structure of phenol red, and in this is a zwitterion in acidic pH. So it gives a yellow color, and at neutral or at physiological pH it's an anion, and it gives a reddish orange color, and at alkaline pH it is a dianion, so it gives a pink or a magenta purple color. So this is how the color change works. Next is uh, other options that were given, which is temperature, growth, and CO2 level. So, temperature phenol red is definitely not involved in any sort of temperature detection. So, phenol red is not temperature sensitive. pH indicators respond to H plus ions and non temperature shifts. And growth of microbial contamination. I wanted to discuss this because microbial contamination CO2 levels are, you can interpret it indirectly, but like it is shown here, it can be an indirect cause depending on the color change, but it's not used for that purpose. The only main purpose is pH change. So that pH change can be again due to CO2 levels or microbial contamination. So it is a secondary interpretation of whatever you have seen in terms of color change. So growth of microbial contamination, if bacteria or fungi contaminate the cell culture, they metabolize nutrients and release acidic products. So because they grow in huge number, right, they proliferate much faster than our mammalian cells. So they multiply exponentially, and then they use the nutrients also exponentially, and then they release more acidic products when compared to our normal cells in a relatively quicker time. So the pH drops immediately as soon as there is contamination within few hours, and it turns yellow in color. So you can indirectly check for contamination. So the phenol red can act as an indirect contamination signal, but this is a secondary and non-specific and could also be due to cell overgrowth. So in the same way, if you are overgrowing your cells, so you basically grow your cells up to only what, like 80% or maximum to 90% confluency. So after that, you either freeze them or you use them for your experiment, or you split them and subculture them. But if you leave your cells for more than the time that it is supposed to be, then it has grown 100%, and then it has overgrown, and it has, instead of being a monolayer, it is like multiple layers now because you have forgotten about the plate. So even that time, due to excess cellular growth, there will be change in the pH and the color changes. Then CO2 levels is also indirect, like I said. So it can indicate a lack of CO2 or high CO2. So CO2 dissolves in the medium, forming carbonic acid, and pH decreases. And if the CO2 concentration in the incubator is too low, the medium becomes alkaline and the media becomes purple. So if too high, it becomes yellow, and if it's too low, it becomes purple. So it is an indirect measure, but basically the change is because of pH. So again, phenol red doesn't actually measure the CO2, but indirectly gives an estimate of what could be the level of CO2, whether it's less or more. So this is also could be a potential reason. It's not sure that that is the reason. So that is why all these options are not correct, and it's only the pH. So next is the serum containing medium. So what is serum and what is a medium without serum? So serum is basically the blood serum that is extracted, and we usually use FBS, which is fetal bovine serum. It's extracted from calf, and uh, because we still do not know like what are all the components present in the serum, so we cannot supply everything in chemical form to the in vitro cell culture in the media. So we need the serum components, either the growth factors or any kind of minerals, vitamins, trace elements, attachment factors, or proteins, etc., which are present in the body. We do not really know how much or what are all the components present in a serum. So unless you give proper physiological condition to the cells, they will not mimic how they are represented in the body in the same way in your cell culture. So for that reason, we have to add serum to the cells during their growth. So for proper healthy physiological growth of the cells, we add something called as FBS, fetal bovine serum. So the basal medium like DMEM, RPMI, MEM, etc. are usually supplied without the serum, and serum is externally supplemented to the media before using it. So typically 5 to 20% serum is used depending on what type of cell you're using and what kind of experiments you're doing. So usually a standard maintained for regular proliferation and growth of mammalian cells is 10% FPS. If your cells need more serum, you can go up to 20%, or if you want to do certain studies where you do not want the interference of serum, you can decrease the concentration to 5%. And sometimes serum starvation is given to the cells to either, you know, stop their proliferation or either to uptake certain elements which might uh, not be efficiently taken up if there is serum present, because a few factors of the serum could interfere with certain drugs or certain hormones, etc. So that time you can even starve yourselves of serum for a short period of time only during the treatment. So that can be done. So the percentage is based on your experiment, your growth, and the cell type. So, what serum provides? Serum provides growth factors and hormones, stimulate cell proliferation and differentiation, attachment factors like fibronectin, vitronectin, and help in anchorage dependent cells to stick to the plastic surface. And it also has transport proteins like albumin, transferrin, which carry lipids, metals, hormones, and nutrients and metabolites like amino acids, peptides, vitamins, etc. It also has protective molecules which can neutralize toxins, buffer against pH or oxidative stress. So these are all the components of the serum. The advantage is that it is easy to use, supports a wide range of cells. It provides a forgiving environment for beginners. So when you don't know what exactly is required and when you're just beginning the cell culture, even if you have forgotten to add any other component, or if you're not sure of all the components in the media, in case you're preparing a media, so serum supplement will somehow manage it to a certain point. So basically it helps in all ways possible for your cells to survive better. Then disadvantages is its undefined composition. So like I said, we do not know in every set of serum what are all the components present, because every time every batch comes from a different animal, every batch is produced at a different time, the extraction and the source is not always the uh, like a constant uh, defined source. So from animal to animal, there could be differences. So we cannot really say that every batch will be exactly the same. There might be some differences. Sometimes it so happens that there could be a batch of serum which is not good, or if it's not effective, then it will also hinder the growth of your cells. So this is a disadvantage, that it is undefined composition, and sometimes there is risk of contamination. Since it is extracted from animal source, from the calf, so it is if the calf is infected with any kind of disease, or if it was undetected, it was not checked properly, or it was a carrier of something, it was not in a detectable range, sometimes there is a risk of contamination from viruses, mycoplasma, prions, etc. And then definitely there is ethical concerns, because it is derived from fetal, it's extracted from the blood of the fetal bovine, that is the calf. So there are also ethical concerns. So it's very expensive for that reason, because it's not very easy to extract. And the source is obviously an animal, and when you need it, you need it in huge amounts. Then it also sometimes interferes with the downstream biochemical assays. Serum proteins can mask cell secreted proteins. So, like I said, sometimes whether your drug treatment or your hormones, or if you're working with any kind of proteins and you want to check something in the media, so that time you starve your cells with serum free media so that you can do your experiment without any interference from the serum proteins. So there could be some sort of serum protein interaction, or serum protein might mask some of the things which you might want to detect, which is pertaining to your cells. So in that cases, we can always go to 1% serum, or sometimes even completely no serum media also. So that no serum media is called as serum free media. So it is the basal media with defined supplements, that is whatever media that is given to you by the company, it will have all known compartments. So serum is the only one which has some undefined components, so you do not have anything of that sort in a serum free media. So, if you want to use that continuously for growth, if it's for a shorter period of time, you can directly add the basal media. But if you want to use it for the growth and maintenance of the cells, then you have to supplement other things externally. So, supplements like specific growth factors, EGF, FGF, PDGF, insulin transferrin, all this must be added. And then defined lipids, vitamins, trace elements must be added. Synthetic attachment factors must be added, or coatings must be done on the plates with poly D lysine, collagen, laminin, etc. Otherwise, you cannot maintain your cells for a longer period of time. So, what are the advantages of doing the cell culture in this way? Is it is chemically defined, you know all the components that you're using in that, and the results will be highly reproducible because there is no change. Every time you can add the exact same amount of each component that you are using. So it will be constant, there will be no variability in your results. And it's safer, there is reduced risk of contamination, and it's better for studying secreted protein signaling pathways and proteomics. However, it is more expensive because you have to buy individually all these elements. These are only a few that have been mentioned here, but there are so many other elements present in the serum which are required for the cellular growth. So, purchasing everything individually for a in a huge quantity for maintenance of cell culture is very, very, very expensive, and it requires optimization for each cell type, and not all cells thrive in a serum free media. So some cells which are sensitive and finicky, they might not survive well in such kind of media. Some cells grow more slowly compared to others in the serum containing conditions. So it's also, it retards the growth because maybe it's not like optimal, and because you have to standardize. So that is why we go with the fetal bovine serum. So the main difference is the presence or absence of growth factors and other undefined serum components. So serum media is natural, defined, undefined source of growth factors. Serum free media must be supplemented with defined growth factors for survival and proliferation. So serum containing media has growth factors supplied in an undefined source, and it has nutrients, amino acids, glucose, ions, etc. It has buffering salts, it has phenol red, etc. And reproducibility is variable because the batch to batch variation will be there, and then the cost is relatively cheap when compared to serum free media in which you have to supply all the growth factors and all the other components. Contamination risk is, however, higher, and it is regularly used in cell culture and primary cultures. Then, serum free media added as defined supplements. It is not, all these things are not present in it, but you need them for continual continuous growth. So you have to add these. This is the only difference. Then other than this, nutrients like glucose, amino acids, salts, phenol red are already present. Reproducibility is, however, high because everything is defined and everything is same. There is no batch to batch variability, and cost is very expensive because you have to purchase each component individually, and then contamination risk is low, and so it is used for more specialized assays and proteomics and other things where your serum factors might somehow or some way could inhibit your experimental setup. So this is regarding the serum containing media and serum free media. So next is preparation. During the preparation of cell culture media, why don't we autoclave the media? So like we discussed, there are so many nutrients and growth factors in the media. So media, like I also mentioned, media can also be purchased as a whole from companies, or you can prepare the media you by dissolving all the components or by dissolving a composite media powder, and then you have to sterilize it. So you cannot put it in UV, you cannot autoclave it because it will degrade all the nutrients. So you have to filter it. So autoclaving is generally done at 121 degrees Celsius at 15 psi pressure for 15 to 20 minutes. So at this temperature and pressure, your macro and micronutrients will be destroyed. So this this is this these conditions are only excellent for sterilizing simple heat stable solutions. Like if you have any kind of buffers like salt, in which you have added only salts, you can just heat it because it won't degrade the salts. Any kind of protein addition in the solutions, any kind of nutrients, amino acids, these kind of biomolecules will get degraded. So mammalian cell culture media are chemically complex and are heat labile. So autoclaving would degrade the amino acids, would degrade the vitamins, proteins, growth factors, carbohydrates, and because it degrades all these things, it will basically change the buffering properties of it, because it also destabilizes the bicarbonate buffer system. So medium becomes non-supportive and even toxic for the cells. So if you use an autoclaved media, your cells might not survive. So for example, L-glutamine breaks down into toxic ammonia at high temperature. Vitamin B complex and ascorbic acid are heat sensitive. Serum proteins, insulin, transferrin, AGF lose activity. Glucose undergoes caramelization. Obviously, if you heat sugars like glucose, it will undergo caramelization at such high temperature. So that's why the color change will also be visible for being inhibitory byproducts. So that is why we never overlaid cell culture media. So instead, filter sterilize them. So we use syringe filters, or we use uh, uh, 0.22 micrometer pore size filters, which will help remove the risk of contamination from bacteria and fungus. So instead of autoclaving, media are sterilized by using membrane filters, commonly 0.22 micrometer pore size. This removes bacteria and fungi. It also preserves the nutrient integrity since no heat is applied. Keeps growth factors, hormones, and vitamins biologically active. Filtration does not remove viruses and mycoplasma, however. So, viruses and mycoplasma are much smaller in size, and they cannot be removed by filtration. So, the filtration only helps till bacterial and fungal to remove the bacterial and fungal contamination. So that is why it is also done in aseptic conditions, which you do it in a UV sterilized room and in a hood which has HEPA filters, so that it has filtered air already coming in, and in such a region you will generally not expect any contamination from virus and mycoplasma. So it has to be done in aseptic conditions, and it has to be filtered using 0.22 micrometer pore size membrane filters. So components like PBS, you can autoclave, like I said, the buffers in which you add only salts can be autoclaved because salts are heat stable. So PBS has only like phosphate buffers, sodium, etc. Basic salts like sodium chloride, potassium chloride, magnesium sulfate, etc., because these are not organic. Glucose solutions is, however, filtered. Like I said, it gets caramelized due to heat and it becomes a byproduct. And then complete medias are all filter sterilized, and serum is also filter sterilized because the heat can destroy the growth factors. Then antibiotics are also filter sterilized because it will lose their activity because they are heat labile. Growth factors, cytokines, hormones, etc. All lose their activity with heat. So all these are also only filter sterilized. Then next is what is present in animal cells but absent in plant cells. That is cilia. So cilia is short hair-like projections on the cell surface made of microtubules which help in cell motility. So movement of cells occur and movement of substances across the cell surface occurs, like in case of the respiratory tract, common in animal cells, absent in higher plant cells. Exception is some lower plant gametes have it, like mosses, ferns, and algae. They have flagella but not cilia. Cilia is generally absent in all the higher plants. And other options are mitochondria. Mitochondria is present in both plant and animal cells because both the cells have to respire and produce energy. So this is present in both. Vacuoles is present in both because vacuole is needed for like storage and other functions uh, um, like pinocytosis or like phagocytosis, etc. And it's not completely absent in animals. Then chromatin is definitely present in both because it's the DNA and it is required for the cell to survive. So this is the table in which what are present. So cilia or flagella is usually absent in plant cells, except for in gametes of some, but often present in animal cells. Cell wall is present in the plant cells, which is made of cellulose usually, and it is absent in the animal cells. Chloroplast is uh, a hallmark of plant cells, photosynthetic plants, and it's not present in animals. And vacuoles are large, and central vacuole is present in the cells, and small temporary vacuoles are present in animal cells. So these vacuoles sometimes also come due to stress conditions, etc., etc. Then centrioles are usually absent in higher plants and present in during the spindle formation in animal cells. Lysosomes are rarely present in plant cells but are definitely present in animal cells because they are used in many clearance processes and autophagy, etc. So, what is an alginate? So, it is natural polysaccharide extracted from brown seaweeds. So, it contains basically M and G units. M is manuronic acid and G is guluronic acid. The special property is it forms gels in the presence of divalent cations like calcium, etc. So, role of alginate in microbiological media. Main function is it is used as a gelling agent. So when it comes to contact with divalent cations, it cross links and forms a stable hydrogel. This gel entraps microorganisms, nutrients, enzymes without killing them, making it useful in specialized application. So why not the other options? Because growth factor, amino acid, energy. So growth factors, as like we know, they are vitamins, hormones, cytokines, etc. And alginate is not a growth factor. It's not an amino acid source. It's not an energy source. Most bacteria cannot metabolize alginate. Only marine bacteria can degrade it. So alginate is basically a gelling agent used, and it's extracted from brown seaweed. So some applications of alginate in microbiology is used to entrap living cells, immobilization of cells and enzymes for fermentation or bioreactor studies, allows reuse of microbial biomass and while maintaining viability, drug delivery and encapsulation used in biotechnology for controlled release of drugs, probiotics and growth factors. It is also used in tissue engineering and it is a biocompatible scaffold for growing mammalian cells. Micro encapsulation in case of cell culture. It's useful in microbiological experiments where cells need to be studied in 3D. So a 3D matrix is made with the help of the gel so that the cells are not in free suspension. It's in a matrix but also in 3D. So this is the source. This is so there is also agar similar to this, which is produced and extracted from algae. So alginate is produced from brown algae, agar is produced from red algae. So both are used for like gel as a gelling agent and for solidification. Alginate requires divalent cations. Agar is, however, thermoreversible. So, agar, we usually heat it, it melts, and then we cool it down, it solidifies. Then used in microbiology for encapsulation, immobilization, specialized media. This is solidifying general purpose of microbial culture media. So we use agar to grow the like plate agar in petri dishes and help grow the bacteria on it. Then biodegradable by some marine microbes. Very few microbes can degrade agar. Strength is softer, porous gel. Agar is much stronger and firmer gel. So alginate is a gelling agent, not a nutrient, and yeah, it's used for encapsulation, immobilization, and biotechnological other applications. Next is other options that were given. Yeah. So Luria Bertani mouth. LB is usually the medium which we use to grow the bacteria. So its composition is tryptone, yeast extract, sodium chloride, and it provides nutrient-rich environment for E. coli, supports fast growth and high cell density, routine cultivation and maintenance of E. coli, preparing cultures for cloning, plasmid amplification, expression, etc. It can also be used for storing the cells by freezing them in glycerol stocks. So it is standard because it is easy to prepare, it's inexpensive, and it works most best for E. coli strains. Whereas M63 medium is a minimal medium. It has glucose, ammonium chloride, and salts. It is a defined medium. It contains only essential nutrients. And it is used for metabolic studies to study auxotrophy, metabolic pathways, etc. Not used for maintenance of growth because it slows the growth of the E. coli when compared to LB medium. SOC medium is similar to LB, but it has a richer component. So it's similar to LB but richer and contains tryptone, yeast extracts, sodium chloride, glucose, magnesium chloride, magnesium sulfate, and KCl. Function is it's super rich recovery medium. So specifically after transformation, after the stress of transformation, when introducing plasmid, the super rich medium helps the cells recover and express antibiotic resistant genes and begin dividing before plating on the selective media. Not used for maintenance since it's costly and unnecessary for routine growth. DMEM is a mammalian cell culture media and it has glucose, amino acids, vitamins, salts, bicarbonate, and often FBS, like we discussed. And its function is used for mammalian cell culture and not for bacteria. It's a cultivation medium for all animal cells like fibroblasts, kidney cells, neurons, etc. Completely unsuitable for bacterial cells like E. coli. So this is a table comparison about all the media options that were given. Then the next is the structure of gram-negative cell wall in bacteria. So the gram-negative cell wall has uh, lipopolysaccharide layer, which is the outermost layer. So the LPS is found on the outer leaflet of the outer membrane, unique to gram-negative bacteria and consists of three parts, which is lipid A, core polysaccharide, and O antigen. So lipid A anchors LPS in the membrane and acts as an endotoxin. Core polysaccharide provides structural stability, and O antigen is highly variable and helps bacteria evade the host immune system. Function is it acts as a permeability barrier and is a virulence factor as well. Then next is the outer membrane. It is asymmetrical bilayer, that is a layer, and outer leaflet has LPS and the inner leaflet has phospholipids. So contains porin proteins and for passive diffusion of small molecules like sugars and ions, and protects bacteria against antibiotics, detergents, and bile salts. Then peptidoglycan layer, a very thin and compared to the gram positive. So peptidoglycan is outermost for the um, gram positive bacteria. It is also present in gram negative, but it's usually thinner than the gram positive. So in gram negative, it is about 2 to 7 nanometers, and in gram positive, it is about 20 to 80 nanometers. And it is sandwiched between the outer and the inner membrane. It is located in the periplasmic space, which also contains enzymes like beta lactamase, degradative enzymes, and transport proteins. It provides shape and mechanical strength but is less rigid than the gram positive. Then inner membrane, it's a classical phospholipid bilayer. It's a cytoplasmic membrane and it contains transport proteins, transport chain components for respiration, anchors cytoskeletal elements and secretion systems. It is selectively permeable barrier separating the cytoplasm from the periplasm. So the order at which all these layers are present is like polysaccharide, outer membrane, peptidoglycan layer, and inner membrane. Coming to lysosomes. So lysosomes, like we discussed previously, are absent in the plant cells. Their role is taken over by a central vacuole in the plants and is related to vesicles. So a vacuole present in the plants for storage of water, ions, pigments, and waste and helps maintain the turgor pressure. Plasmids are also unique to plants and algae. Example, chloroplasts, amyloplasts, and chromoplasts. And they are also specified, specialized organelles which have specific functions like the chloroplast has chlorophyll for photosynthesis, amyloplasts have starch for storage, and chromoplasts have pigments which give color. Then lysosomes are absent in higher plants. Instead, plant cells use their vacuole for breakdown of macromolecules and storage of hydrolytic enzymes. Like structure sometimes exists, but a distinct lysozyme as in an animal cell is generally not present. Then peroxisomes present in both animals and plant cells. In plants, they are important in photorespiration and breakdown of fatty acids. Glyoxysomes in case of germinating seeds. Then next is what are mesosomes? Mesosomes are folded invaginations of the plasma membrane. So the plasma membrane of bacteria folds and it forms an invagination like this. So these are called as mesosomes. These were first observed under the electron microscope in the 50s and 60s. Today they are widely considered as artifacts by chemical fixation techniques, etc. So now they are saying that these invaginations might not be actually present in the cells, but they could be formed during sample preparation. That is, when you use dehydrating agents or when you use staining agents or any fixatives like osmium tetroxide, etc., that might lead into formation of mesosomes. So they are basically considered artifacts. However, in classical microbiology, they are still described with functional importance. So, it is present in bacteria attached to the plasma membrane. They are not free in cytoplasm, not linked to ribosomes, and not part of the cell wall. So there are different types of mesosomes as well. So there are septal mesosomes found near the cell division septum, involved in binary fission, DNA replication, and chromosome distribution. Lateral mesosomes found on the sides of the plasma membrane, believed to be associated with respiration and secretion. And functions, the classical view of mesosome functions are, however, debatable, but mesosomes were thought to play roles such as respiration, DNA replication, cell division, cell wall synthesis, secretion, etc. So, in case of respiration, it is thought that they help in increasing the surface area for the respiratory enzymes in like in mitochondria. And for DNA replication, they provide an attachment site for bacterial chromosome. And in cell division, they help in equal segregation of DNA to the daughter cells. And in case of cell wall synthesis, they provide anchor points for the enzyme involved in peptidoglycan assembly. And in secretion, it helps in release of extracellular enzymes. So the other options, free in cytoplasm, attached to ribosome, attached to cell wall, are all wrong because mesosomes are membrane folds and not cytoplasmic structures. The ribosomes bind to mRNA and not mesosomes. Mesosomes are part of plasma membrane and not the cell wall. So, okay. So there are, I think, two people as of now. So if you guys have any question or any doubt, you can stop and ask me, or you can put it up in the chat box. So we'll continue. So the next is the background of Mitsuhashi Maramorosh medium. It's the medium for mosquitoes. So it was developed in 1964 by Mitsuhashi and Maramorosh, specifically designed to support mosquito cells in vitro, which was difficult before this medium was formulated. So, this helped researchers to propagate mosquito derived viruses like arboviruses without using live insects, enabling safer and more controlled experiments. The composition of this medium is a semi defined medium tailored for insect cell metabolism. So it contains amino acids, vitamins, salts, sugars, and serum, often fetal bovine serum to provide growth factors. Then it is optimized. Uh, Osmotic conditions, pH, and nutrient balance for mosquito cells and supports adherent or suspensions cultures depending on the mosquito cell line that is used. So the primary applications are culture of mosquitoes, mainly Aedes aegypti, vector for dengue, yellow fever, and Zika. Virus propagation of arboviruses such as dengue virus, chikungunya virus, and Zika virus. Vector biology studies to understand the mosquito physiology, gene expression, and host pathogen interactions. This is a table where you can see, like for Drosophila melanogaster, we use Schneider's Drosophila medium, and it is optimized for fruit fly, especially cell lines like S2 and KC. Then for Spodoptera frugiperda, Grace's medium is used for baculovirus expression system, and for Trichoplucia knee, we use TNM FH medium or Grace's medium. Then for Aedes aegypti, we use Matsuhashi Marmorosh medium, and this is specifically for arbovirus research in mosquitoes. Next is coming to the densities. So when using centrifugation to separate components, the densest particle travel the furthest at the tube. So while the least dense particle remains at the top. So when you centrifuge at a lower speed, the higher density particle will pellet down at the bottom of the tube. And as you gradually increase the speed of the centrifuge, the components start to sediment at the bottom of the tube depending on the densities in an decreasing order. So the higher the density, the first it will be sedimented or come out in the pellet, and the least dense will be still in the suspension. So mitochondria, among the other options, are largest and least dense, and their buoyant density is generally in the range of 1.07 to 1.11 in sucrose gradient. And viruses, the density of viruses varies depending on the specific virus and the medium used for centrifugation, but typically the range is 1.14 to 1.24. And DNA is double stranded. The density is typically 1.7 g per centimeter cube, and it is higher than both mitochondria and virus. RNA is densest. The density generally ranges from 1.7 to 2 grams per ml. So the order of density in increasing is mitochondria, virus, DNA, RNA. So this is how you can use this to separate the different organelles, separate the different organelles or different cells depending upon their densities. So you have a cell homogenate here. So at low speed, when you centrifuge them, you will get whole cells, nuclei, and cytoskeleton in the pellet. Then when you increase the speed much higher, you will get mitochondria, chloroplasts, lysosome, peroxisomes in the pellet. Then further, when you increase the speed, you will get microsomes and small vesicles, which are which have very less density, and then the least dense ribosomes and viruses. So viruses, depending on the type of viruses and depending on the medium that is used, they can be least dense or they can be similar to mitochondria also. So this is how the differential centrifugation is used to isolate or fractionate different parts, different organelles from a cell suspension or a cell homogenate. Next is the composition of TSC buffer. So the TSC buffer, this is an abbreviation to the components itself. So T stands for tris, S stands for sucrose, and E stands for EDTA. So tris HCl is used at a pH of around 7 to 8. It maintains stable physiological pH and prevents protein denaturation. Sucrose acts as an osmotic stabilizer, and it is usually used in a concentration of zero point 0.5 to 1 molar, and it prevents cytoplasmic membrane from bursting when the outer membrane is weakened. Then EDTA is a chelating agent, so it chelates divalent cations like magnesium and calcium that stabilize the lipopolysaccharide layer of the outer membrane in case of gram-negative bacteria. It also weakens the outer membrane, making it more permeable. Why TSC is used for periplasmic extraction? Periplasmic space is basically located between the inner and the outer membrane in case of gram-negative bacteria. It contains enzymes like alkaline phosphatase, beta-lactamase, binding proteins, and small molecules. So the mechanism is the EDTA destabilizes the outer membrane. The sucrose maintains the osmotic balance to prevent cytoplasmic lysis. Gentle treatment allows the periplasmic proteins to leak out into the buffer. The cytoplasm remains intact. So this method is sometimes also called as a osmotic shock because the outer membrane is weakened without breaking the cytoplasmic membrane. So this is gist of the protocol, that is the suspension of bacterial cells is put in TSE buffer so that the EDTA weakens the outer membrane, then incubate briefly, the periplasmic enzymes diffuse out, and then centrifuge gently to remove intact cells. The supernatant contains the periplasmic fraction, and then other optional steps are dialysis or concentration of the periplasmic proteins. So it can be used for the extraction of periplasmic proteins without damaging the cytoplasmic membrane. Then, in case of mitochondrial isolation protein purification and cytosolic fractionation, TSC is not used. So in mitochondrial isolation, we need homogenization buffer, and the main components are sucrose and mannitol, and not EDTA. For organelles, cells must be lysed. Here it is the cytoplasmic membrane is kept intact, but however, for mitochondria, the cell has to be completely lysed. Then for protein purification, TSC only extracts the periplasmic protein in case of gram-negative bacteria. But general protein purification is not done using TSC uh, because the cytoplasm still is intact. So the cytosol fractionation requires complete lysis of cells, and TSC is too gentle for this. We go to processes like sonication, French press, or use detergents. So TSC is basically used for periplasmic proteins. Then sterile filtration in case of media. So we discussed sterile filtration instead of autoclaving is done for sterilization. So mammalian cell culture media contains heat sensitive components like amino acids, vitamins, growth factors, serum protein, etc. So like we discussed earlier, autoclaving will denature all these biomolecules and reduce the cell viability or even make it more toxic to the cells. So filter sterilization is the only efficient way. So the filter pore size that is used is very important. Like the 0.22 micrometers filter size is used. It is the one where most bacterial fungal spores are eliminated. So it is standard for sterilizing mammalian cell culture media. Point 2 is similar to this but slightly tighter. That is slightly it has a smaller pore size and is sometimes used if you want extra care and you want to use for more sensitive media. Point zero two is for viruses and nanoparticles and used for virus filtration and not for media. And 2.2 is for large debris excretion, so too large and bacteria can easily pass through this. And 22 microns is very coarse and it is ineffective and like it is basically it does not sterilize the components. So, pore size and its significance. Why 0.22 microns specifically? So, most bacterial cells range between zero point 0.5 to 2 microns in size. So, a 0.22 micron pore size effectively blocks all the bacteria, letting only the nutrients and macromolecules pass through. Fungal spores are slightly larger and also removed. Materials of the fiber filters that is used are generally PES, polyether sulfone, cellulose acetate, nylon, or PVDF. PES has low protein binding, so it preserves the growth factors. And cellulose acetate is widely used for general purpose filtration. Nylon PVDF is used depending upon the chemical compatibility because proteins bind to nylon and PVDF. Then sterile filtration process. Media is prepared in a clean container, autoclaved container, and passed through 0.22 micron membrane filter using a syringe or a vacuum or a pressure filtration system for large volumes. And filtrate is collected in a sterile container. Filtration removes bacteria spores but preserves heat sensitive nutrients. So that is why 0.22 filters are generally used for sterilization of cell culture media. So this brings us to the end of this session, I guess. Yeah. So we are done. I hope we have covered all the questions. So if you guys have any doubt, you can ask me or you can leave the meeting. We can end the session.